Hello everyone. So I hope I'm audible, and so we gonna start in 5 minutes, okay? So that people can join in. Otherwise, they trickle in between the session, and it creates unnecessary disturbance for you as well as for me. Okay? So we'll just wait for 5 minutes.
disgrace poaching county from outsider Dostoevsky की कहानी का विषय ऐसे और भी कई अलग
Yeah, so as we wait for other ma'ams, hi ma'am, today's class we did not have problem solving. Um, Janki, what do you mean we don't have problem solving session? Uh, Either you can unmute yourself or you can type in the text box. Do you mean that we have not solved questions or something like that? Yeah, so just a little update. So I came to know that the meeting link which I have created via Google, this Google Meets, the thing is, it only permits me to hold the session for 1 hour. Okay, that is why the last time the meeting got suspended on its own. So I was not aware of this while I was creating the link, because last year I used this Meets platform itself, and at that time I didn't face any such issues. So maybe this time as well, what will happen is after 1 hour the meeting will automatically get suspended. So I will be still there. I'll rejoin the link, and you can also join via the same link, and we'll carry on from there itself. Okay, so I'm so sorry for this, but as I said, I have used the same platform last year and I didn't face any issues last year, because I was able to conduct sessions even for like lectures for 2.5 hours, 3 hours continuously as well. So, and if I change the link right now, first of all, I won't be able to change it, because it will be an unnecessary chaos for the people ki, oh, this is the new meeting link, then they have to come, so unnecessary confusion will be there. So just a request, if the meeting, it will definitely get suspended after 1 hour, so you just rejoin the session. Okay, again I'll wait for 3 to 5 minutes once I restart the session, so that you guys don't miss anything in between, and I'll put the same thing in the chat as well.
Good afternoon, ma'am.
uh, yeah, hi.
Ma'am, last class I was missed. There will be recorded available, ma'am.
I have
Session.
not uploaded the last session till now, but yes, the session are being recorded and they'll be uploaded on YouTube.
Okay, ma'am.
Yeah,
Thank you, ma'am.
yeah, no problem. Okay, I have removed the permission so that people can just join in. I don't know why it is still asking me to let people in. Yeah. I guess we can start, and as and when people come, we'll just let them in, and I hope that it doesn't ask for permission again and again. So just let me know whether you can see my slides and I'm audible. I'm assuming I'm audible because I just interacted with the student. So yeah, I think we can start our today's session. So hello, good evening everyone, and I apologize because I have kept my sessions in Sunday evening, but that was the only slot which was available to me as well. I apologize for that. So in last class what we have discussed is it's just basically the overview of immunity. Okay, we try to understand what is immunity, what is immune system, what are the players in the immunity, right? So all these things we were seeing, and then gradually we went into types of immunity, right, or types of immune systems, or the participants which are present in these different types of immunity. So the first thing first is that till now, as we are already in the second week of our classes, you guys are very well versed with the terminologies, right? That which type of cell is associated with which type of immunity. So in the last class we just skipped through the innate immunity. Then we started discussing about the adaptive immunity. Then in the adaptive immunity itself, we saw that there are two arms of adaptive immunity, right? First was the humoral branch and the second was the cell mediated branch. So in humoral, we know what sort of players are involved, and in cell mediated, what sort of players are involved. So if you guys have followed the week's lecture which professors have uploaded in the NPTEL platform itself, then following these sessions, as I've told since the first day, won't be difficult. But if you guys are not following or keeping up with the pace of those lectures, then things might become little bit of murky at the end of the day. Okay, so instead of following what is there in the lectures, because you are already learning from the lectures, right, the video sessions which are uploaded there. So what I am trying to do is the same topics and how we can understand them in an alternate light. It's the same thing but the alternate light. Okay, so moving forward from there, so as I have mentioned in the last class, we were discussing for the adaptive immunity. So till last class we talked about humoral immunity and cell mediated immunity, correct? Now we'll see that you can always like, okay, I don't know why people are not getting admitted automatically. Okay, leave. Okay, so last time we divided adaptive immunity on the basis of where the players are present. If the players are present or acting via the blood, right, and they need like secretory type of thing, then we categorize them into as humoral immunity. But when the cell is directly involved in attacking or providing the defense, then we called it cell mediated immunity. Now the adaptive immunity can also be distinguished into two different type of immunity. Again, it is not another branch of adaptive immunity. No, it is just a way how we are seeing the adaptive immunity. Okay, so for example, if we say we can divide the living system into two parts, right? Living thing and non-living thing. These are the things we have. Now further, the living things can be divided into plants, animalia and all those things, right? And even further, animalia can be divided further into multiple things. But the moment we change the criteria on which we are dividing, things become different. Okay, so now we are seeing the adaptive immunity from an alternative lens, through which it is divided into active immunity, that is active adaptive immunity, and passive active immunity. Okay, so in active adaptive immunity, it is developed after exposure to a microbe or its antigens. Individual mounts an immune response and eliminates infection. So this is the major type of adaptive immunity which we are familiar with, right? We say once the pathogen enters our body or something happens, our adaptive immune branch is activated. So which branch of adaptive immunity is activated? Is the active immunity, which starts when the microbe enters or when our system sees the microbe. Okay, then provides long lasting resistance, as we know, because there are memory cells associated with it, right? Then exposed individual becomes immune, that is the whole concept of vaccination, right? Then unexposed individuals are considered naive. Why? Because they won't have the antibodies, they won't have the memory cells, because their system have not seen the antigen, right? This is the branch which we majorly deals in when we say that we have vaccinated the population. Okay, because we have shown the system that, oh, see, this is an antigen which might come from the outside, so you should be prepared beforehand. So this is the branch which we are dealing with majorly when we are talking about, again, vaccines. Okay, another branch of adaptive immunity is called passive adaptive immunity. So in passive adaptive immunity is antibodies are transferred from an immune individual or produced in the labs. Okay, produced in the labs, provides immediate but short term protection, does not induce immunologic memory. Why? Because we have not shown the antigen to the body. It's basically what happens when we are imagine you got a snake bite. So now you have to put into an go to the hospital and get an injection of anti-venom injection, something like that. Okay, so in anti-venom injection, you basically have antibodies which were prepared outside, and we are directly delivering those antibodies in your body. But did your body produced those antibodies after seeing the snake venom? No, right? We are directly providing the antibody, hence your body won't produce memory associated with it. So this branch of adaptive immunity is called passive adaptive immunity. Okay, so some physiological examples for the passive adaptive immunity and in general adaptive immunity is newborns receive maternal antibodies via placenta and via breast milk. So we are aware that a new baby is born. So the new baby will have the innate immunity, right? Because it is inherent. So it is developed by the baby by its own system. But the moment a small baby comes out into the world, lot of antigens are present around it, right? Yet babies still survive. They do not fall sick that often. Why? Because they have antibodies in them without seeing the antigen, and these antibodies are provided to them by the placenta. So there are certain antibodies which delivered to the baby via the placenta during when the baby was inside the womb, and via the breast milk as well. Okay, and then the clinical applications include immunodeficiency disorders. They can be treated with antibodies. So imagine an individual is immunodeficient, so the name itself means that its body is not sufficient in producing the enough immune response against the infection. So what we can do in this case is we can make antibodies outside his body and then give it to the concerned individual. By this we can curb the disease and help the individual survive. Second, emergency treatment of infections such as SARS-CoV-2 and snake bites, as I have mentioned already. Okay. Then monoclonal antibodies used in cancer immunotherapy. T-cell transfer is not feasible between unrelated individual due to rejection. This we'll see maybe in the third or fourth week, how we can say that the T cells cannot be transferred between two individuals. There has to be some gene rearrangement concept needs to be taken care of here. Okay, so till now adaptive immunity was, we saw this in two types. In first type we classified adaptive immunity into humoral and cell mediated. In another way we classified adaptive immunity as active adaptive immunity and passive adaptive immunity. Now what are the responses and the properties of adaptive immune response or the adaptive immunity? So some features such as specificity, that is ensure that immune responses combat the pathogen or
Ma'am,
yeah
sorry for
it's okay
sorry for the interpretation. Ma'am.
yeah.
If maternal antibody protects newborns, why do babies still need vaccines in the early months, ma'am?
Why does the baby, can you pardon your
Need-
question?
Need vaccines in
Okay.
the early months.
Okay, because the maternal antibodies are not sufficient. So imagine you have lived for 20 plus years now. Okay, so your body has seen n number of or n amount of microbes already which are there, and the memory cells are there in your body. So you are not falling sick on daily basis. You don't get cold and cough every season, right? Because you have seen such pathogens, correct? But the baby is a newborn. For him, every single antigen is a new antigen. So these antibodies are not sufficient for the baby to survive and not fall sick like every single day. These are just enough so that the baby can be protected from general general antigens. Now the vaccines which we get, for example, if you are getting though now we don't get polio vaccine or smallpox vaccine or measles vaccines, but these are antigens which are not commonly found in the environment. But as you have been vaccinated, you have those antibodies, but your baby, because that particular antibody is not has not crossed the placenta or has not transferred via the breast milk, so the baby still needs to see those antigens to make antibodies for them, because here not all the antibodies which the mother has is being transported. These are very limited numbers and only a specific type of antibody which is able to cross the placental as well as the breast milk barrier or the physiological barrier. Okay, so just to make sure that the baby wholesome is protected, we still need the baby to be vaccinated. So as we were talking about the properties, so the specificity, so ensures that immune responses combat the pathogens or tumors that are encountered. Diversity enables the immune system to respond to a large variety of antigens. Memory leads to enhanced responses to repeated exposures to the same antigen. Non-reactivity to self, that is, it prevents injury to the host during response to foreign antigens. I guess they are self-explanatory in their own way. So if we talk about specificity, which means whenever the adaptive immune branch of the immunity is activated, it will be very specific. It will only kill the microbe for which it is being activated. Diversity. So the adaptive immune system is very diverse. It can identify multiple types of antigens or say PAMs and can act against the microbe. Then the memory. Once the infection has taken place, the adaptive immune system keeps the memory of the infection, so that if in future same infection comes to your body, either the infection won't be able to sustain, or even if the infection is sustaining, its effect will be reduced. It won't be that severe as it was before. Okay. And then non-reactivity to the self. So every cell, whatever we are talking about, it's part of our own system, right? So once the adaptive part of immunity is activated, this branch of immune response should be clearly able to distinguish whether the cell against which it is acting is a self or from the same host, or the one which is from the other host, or say it's basically a foreign. So the immune system should have these basic properties associated with it. Okay. So as we have seen earlier, like how basically the innate immune response, they have the effector molecules. Similar thing we're gonna see how the adaptive immune system is propagated at the end of the day. So instead of going too much detail into like what are these cell types, because you have a basic idea about them, right? So here I want to focus on how basically the adaptive immune system is providing the immunity. Okay. So imagine there was this lymphocyte precursor, correct? So this lymphocyte precursor cell, what it did is, it was just sitting there. There is some problem with these two individuals. Okay. Yeah, I'm sorry for the disturbance. Yeah, so now we can continue. So yeah, so once the lymphocyte precursor cell is there, so what basically happens is that lymphocyte clones with diverse receptors arise in generative lymphoid organs. This slide can only be understood if you guys have gone through this week's lecture. So could you tell me how many of you have gone through the lectures of this week? Otherwise I can explain the lymphoid tissue and the germinal centers, because without that information this slide is little bit difficult to understand. So raise your hand if you guys have seen the lectures of this week. Not necessarily have completed it, but at least know what is there in the lectures. So kindly raise your hands. Okay. One person is there. Okay, so basically majority of you guys have not seen the lectures, right? So let me check if I have a slide related to this is the inflammatory response. Okay, because I thought you guys are following the lecture, so I don't have that particular topic with me for now. Okay, let's take it like this. So here what we are trying to understand. So we are trying to understand that an antigen has entered in our body. Okay. Now in this case the innate immune system was not able to curb the infection, because when we are getting infection, it is not that we'll get only 1 microbe, 2 microbes, something like this, right? We'll get n number of microbes all of a sudden. So majority of it is dealt with the innate immune system. But another part of innate immune system is that it should tell the adaptive arm of the immunity that, see, this sort of infection entered in our body. Okay, so once the innate system does that, then what happens is how the adaptive immune system reacts to this new information. So what they do is, as by now you know that what is being identified, the PAMs are being identified. PAMs are on the antigen, okay, and then the PRRs are on us, like our cells basically, okay. So once the PAMs are being recognized, they are taken to the adaptive immune system by dendritic cells, and then the adaptive immune system says, okay, let me see what sort of antigen you have brought me, okay. Then they say, oh, they have this type of covering on them, so I have this cluster of cells which are already there, so this is just combination, this is just trial and error, okay. So A, B, C, D, E, these types of cells are already present in our system, okay. This is the back of it, so I'm not going there because you guys have not gone through the lecture, so it will be a little bit difficult to summarize in this particular slide. So imagine you have a bag which has n number of colors of ball, so you have red ball, pink ball, black ball, white ball, all those balls are there in that bag. Now imagine I am asking you to pick only the pink ball, okay. So you will put your bag in that hand and you can't see it, right? So you will pick some random balls, so the chances of you picking the pink ball is equivalent to the probability of the total number or the sum of balls which are present in that bag, right? Similarly, imagine there is a secondary lymphoid organ where different types of lymphocytes are present, okay. So that lymphocyte will be picked further, which can bind to the antigen which has just entered, okay. So imagine this pink ball can be bound to an antigen of X type, okay. This blue cell can be bound to an antigen which is of Y type, okay. Now for us right now, this orange, yellow and green, these cells are not necessary and not needed right now. What is needed? Because X antigen has entered in our system, this is what innate immune system has told us, right? And Y sort of infection is already there. So what will happen once the X antigen binds to its own type of lymphocyte cell, this cell will further divide and increase in its number, okay. Same thing will be done for another antigen, okay. Now what will happen once these cells are divided, because they have been selected, so you can read here, antigen specific clones are activated or selected, right? So this cell is not dividing, this cell is not dividing, as well as this cell is not dividing. Why? Because there is no antigen presented to these cells which can bound to them. Only those cells will be divided further or activated which have seen the antigen and which can bind to it. Once they are bound, then these antibodies are released into the system by these cells, because this is the type of antibody which recognized X. So these antibodies will be secreted in the system and further they can nullify the infection. Same thing can be done for the Y infection as well. Now imagine if there was A type of infection. So in A type of infection, this cell won't work. This type of cell will not be selected for A infection. This type of cell will also not be selected for the A type of infection. But imagine A type of antigen is recognized by this green cell. Now this green cell will be further divided and it will then lead to the secretion of antibodies into the system, which will bind to that A antigen and finally nullify it. Okay, this is the easiest way we can understand clonal selection. And why we are saying clonal selection? Because there are n number of clones which are already present in the lymphoid tissue, but only those clones will be selected which can identify and bind with the antigen which is presented to the adaptive immunity by the innate immunity. And who are presenting these? The antigen presenting cells. Okay, so now you can connect how things are connected in the immunity. Okay, moving forward. Yeah.
Ma'am, can you please explain the clonal selection once more?
Okay.
Means
Say. Continue.
Ma'am, that after the balls are selected, after what you were saying,
Acha.
why that green cell is not means why the green cell at the last is also producing antibodies?
Okay. So green cell is not producing antibody in this case. Why? Because this, so you can see the type of receptor is different here, right? So see this, okay, let me take a pen, avoid my drawing. Okay, so see, what is the shape of this antibody? It's U shape, right? What is the shape of this antibody? It's V shape. Okay, what is the shape of this antibody? It is some random shape like this. Okay, so this microbe has an inverted U shape, which can be easily identified by this type of receptor on the cell. This will not be recognized by this type of receptor. Do you understand this? So this shape, do you think that it will be able to bind with this M type of shape here?
No, ma'am.
No, right? Will this shape be identified by this square type of receptor here?
No, ma'am.
Right? So do we need these cells further?
No.
So it will be equivalent to, for example, you are having a bacterial infection. Okay, you got some wound, you fell into a mud, you got some rashes, and there you have loads of bacteria. So now that bacteria is of inverted U shape. It has PRR of inverted U shape. Correct? So in that case, the antibodies which have the shape, complementary shape of inverted basic U, will be helpful in reducing the number of that particular bacteria. If I have an antibody which is M shape, it is not useful for reducing the number of that particular bacteria, right? But this M shape antibody is helpful if there is an antigen, say some antigen is there, and they have they have this type of PRRs on them. Okay, as I have already said, avoid my drawing skills. Okay, so imagine if this is an bacteria which has this type of surface receptors or PAMs on them. Okay, PAMs you should remember from the first class. What are PAMs? PAMs are pathogen associated membrane. Say okay, you should all of you should revise your lessons. So PAM. So imagine now I have an antigen which on its surface it has M sort of something. It's wearing it's wearing clothes which has M on it. So now this particular bacteria will only be able to bind with a cell which has receptors like this. In that case, I don't need antibodies which are of U shape. I need antibodies which are of M shape. Okay, so the clonal selection basically means with the number and the availability of clones which are present here in the lymphoid tissue, only that clone will be selected which is complementary or able to bind with that antigen. In this case, if we are seeing like whatever is present in this example, in this example only this type of pink cell will produce antibody, because this was the antibody which was able to identify the antigen. The green type of antibody will not be selected because it didn't see that antigen. Maybe in future this type of antigen will come on which the surface of that microbe will have receptors which are complementary to this particular receptor. In that case only this cell will be selected, which means this clone will be selected and this type of antibody will be released. But in this case which we are seeing right now, for antigen X, only this clone is selected because they were the complementary here and they can identify each other. for antigen Y. So this is different antigen, right? So example, this is your cold and flu virus, okay, and this is your, say, TB virus, okay, or TB microbe at the end of the day. So for cold and cough, you won't need any TB microbial antibodies, right? And for TB microbial antibodies, you don't need any bacterial antibody. So this is the basis of clonal selection, that we are only allowing the cells to divide, those clones to divide, when they have seen the antigen and they can recognize it. And what do you mean by recognizing? By recognizing, we mean they can bind to each other, okay. I hope it is little bit clear now, and if it is not clear, then we can again go back to these concepts. But before this, I'll suggest you to go through the lessons one more time, and then we can discuss it further, okay.
Ma'am, so the green cells is not showing clonal selection,
Huh,
but
they are
the
not.
pink and blue one is.
Because they have seen this type of antigen. For it to divide and release antibody, it needs to see an antigen which is of this shape. So you can distinguish. So this is X, you can see, right? It's different looking from the Y antigen. Similarly, here A antigen will come which can bind to this. Once that antigen binds to this, then this clone will be selected.
Okay, ma'am.
So some other features of adaptive immunity are as follows. Adaptive immune responses have other characteristics that are important for their functions. Immune responses are specialized, and different responses are designed to defend best against different kinds of microbes and at different sites of infection. For example, there are several classes of secreted antibodies, each of which perform a different set of functions. And there are several subsets of T cells, each of which combat infections in different ways. All immune responses are self-limited and decline as the infection is eliminated, allowing the system to return to a resting state, that is the homeostasis, prepared to respond to another infection. So what are the other features of adaptive immunity apart from specificity, diversity, memory and non-reactivity against self? Is that there are n number of antibodies, okay, and we'll see this in further classes as well, that what I mean by n number of antibodies. So this is just the representation. Here you can see, so there are 1 2 3 4 5 possible ways of antibodies can be there in your system, but at given time not all 5 of them are there in the system, right? Which means there are several classes of secreted antibodies which are present in your system most of the time fighting the infection. Other thing is, now imagine, so these antibodies were secreted, right? These antibodies were secreted. They have curbed the this infection. So this microbe is gone now. We have digested it, we have reduced it, the microbe is not there in our system. So do we need such antibodies again in our system? No. So we want our body to come back to the level 0 when there was no infection. So the adaptive immunity also excuse me, also plays role in maintaining the homeostasis. So once the infection is gone, all these antibodies or whatever the system defense system was there, it will be mitigated and will be brought back to the 0 or the initial state, which is called the homeostasis, okay. So these, as we have seen for the innate immune response or innate immunity, these are the cells which are associated with the adaptive immunity. So what are these cell types? Lymphocyte. So there are two basic types of lymphocytes: B lymphocytes, T lymphocytes, okay. What are the functions of these B and T lymphocytes? So specific recognition of antigens and generation of adaptive immune responses. So obviously if we are talking about adaptive immunity, so these cells will generate adaptive immune response. But what are these immune responses? B lymphocytes, they are the mediators of humoral immunity, Now you are try to connect with the adaptive immunity itself. What was the humoral immunity? Humoral immunity was when things are secreted into the blood to reach to the antigen and reduce its number or basically take care of it. So what cells are there in humoral immunity from the adaptive immune system? Those are B lymphocytes. Why? Because B lymphocytes are secreting the antibody which we have just seen. T lymphocytes, they are the mediators of cell mediated immunity. That is when the cell itself going to the microbe and killing the microbe. Okay, so that is dealt with T lymphocyte. Now other cells which are involved in adaptive immune system is myeloid cells. So different types of myeloid cells are there. So neutrophils, macrophages, dendritic cells, mast cells. So these are the cells of myeloid origin. So what they do? So neutrophils and monocytes or macrophages, they phagocytosis, and they are involved in phagocytosis and killing of the microbe. So we have seen since the beginning, right? So once the infection is there, the macrophage will go to the microbe, digest it and present it. Neutrophils will also try to engulf it as soon as possible. Okay, so neutrophils are involved in phagocytosis. Dendritic cells, that is antigen presentation to T cells and initiation of T cell response. So we'll see this in the further slides how this is being dealt with. And mast cells, they basically secrete the inflammatory mediators. So if you guys have gone through the lecture, you know that we have discussed the inflammatory system in quite a detail, how basically the inflammation works. Okay, so in that the mast cells plays a major role in initiating the inflammation, as well as they are the end products of the inflammatory pathway. Okay, so I'll discuss this after some time, because I feel that you guys are not prepared for it to understand right now. I'll give a very brief review of this, but we'll get back to this particular graph because this is an important graph in next week's class, because by then you will be exposed to both how the innate immune system works as well as how the adaptive immune system works. Okay, but the brief overview of this is, and remember this is important. Okay, so now imagine that our body has some infection, or say the season is changing in here, right? So we have, sometimes we have cold, sometimes we have fever, and all those things are there, right? So what happen is, once we have an infection, okay, so in that case, that infection, say it is of pink type. Okay, so this pink type of infection is we encountered on this 0th day. So 0th day we have this antigen X. So what will happen? So you know, once this infection enters our body, the innate immune system will be activated and it will roam here and there in searching of this antigen. Now once this antigen is recognized and is presented to the adaptive system, then what will happen? The adaptive system will basically try to secrete antibodies by clonal selection. Okay, give me 1 minute, this is a little bit of disturbance. Okay, yeah. Then okay, so the adaptive immune system via the clonal selection will secrete antibody. So these antibodies will be of what type? They will be of X type. Anti-X type of antibodies will be there. Okay, so imagine on 0th day your system saw antigen X entered. So what will happen in the first response, because this is the very first time our body is seeing X antigen. So what will happen is, during this, gradually the number of antibody will increase, and these antibodies are type of anti-X antibody. Okay, it will increase, and once the infection is reduced to the manageable scale, again the number of antibody will decrease. Okay, and it will maintain some basal level, because the infection was not cured completely, but it was came down to a manageable number. Okay, now here what will happen again is, now imagine in future, maybe say day 5th, you got the same antigen again. Here imagine antigen X, abhi don't see any Y here, okay, just see X, just follow the X. So on 0th day you got antigen X. After first or second day of infection you had maximum antibody against it, and then gradually by 3rd 4th day your the numbers were mitigated and you have memory B cells. Now your system knows that antigen X had arrived your body and is being dealt with. Okay, now once again due to some reason again you got the antigen X on 5th day. Now what will happen, if this infection was new, it will again follow this trend, that it will again learn, the clonal selection will take place, the antibodies will be released, right? But now what will happen, once the antigen X enters again, we already had memory B cells against this antigen X. Now suddenly, because we don't have to follow further steps which were there in the primary infection, now with the help of memory cells and the memory against the X antigen, the number of antibodies will be extremely high within 1 day, as you can see here, right? It took at least 2 to 3 days here. Here within 1 day the number of antibody against antigen X will be too much into the system because we had memory earlier, right? So with this the antigen will be dealt with very strictly, very specifically, and later gradually the infection load will be reduced. Okay, so this we only followed antigen X. But once your skin is exposed, or say you are exposed to some type of infection, you won't only get one microbe, right, or only one type of microbe, correct? So imagine now, once your system was seeing antigen X again, so on 0th day we had antigen X, on 5th day again we had antigen X, but with this now you went to some place and you got antigen Y as well, right? But now for antigen Y, your body is seeing this Y for the very first time. So what it will do, it will activate the adaptive immunity, then the clonal selection of Y type will happen, and then again it will curb the infection. But this will be the primary response for antigen Y, and at the same time if you got again antigen X, it will be a secondary response for antigen X. Why? Because primary response to antigen X was already there at 0th day. Okay, I know this is a little bit of confusing because either this is the first time you are seeing this graph or you have not followed the sessions. So I'll highly request every one of you, please go through the lectures and we'll explain and we'll visit this graph again. Okay, because this is the basis of immunology and the basis of immunity at the end of the day. Okay, yeah, moving forward. So okay, so these are basically okay. So let me so as you guys can see in the chat itself that it is about to be 1 hour. The session will suspend on its own. So what I'll do is I'll suspend the session because we are entering into problem solving and that type of thing. So please all of you rejoin the meeting from the same link. Okay, because there is some time restriction with this particular link which I was not aware before creating the link. So I'm gonna end this meeting right now, but immediately all of you join this meeting. Okay, please just raise hand or say okay. Okay, you understood, right? Okay. Okay, so you guys can see my screen. I'm assuming that. So before going to the questions and giving you more terminologies and more numbers, let's have a basic view of things what we have learned till now, what cells which are already involved, and what sort of things we can expect. For that, this was a very basic video which I could find. So just let me know whether you guys can hear the like audio of it. Okay. Can you hear the audio?
Yes, ma'am.
Confirm me one more time, okay?
Being surrounded by harmful organisms, toxins, and the threat of our own.
We can hear.
Yes, ma'am.
Oh, okay. Thank you. Great.
Despite being surrounded by harmful organisms, toxins, and the threat of our own cells turning into tumor cells, humans manage to survive, thanks largely to our immune system. The immune system is made up of organs, tissues, cells, and molecules that all work together to generate an immune response that protects us from microorganisms, removes toxins, and destroys tumor cells. Hopefully, though, not all at once. The immune response can identify a threat, mount an attack, eliminate a pathogen, and develop mechanisms to remember the offender in case you encounter it again, all within 10 days. In some cases, like if the pathogen is particularly stubborn, or if the immune system starts attacking something it shouldn't, like your own tissue, it can last much longer, for months to years, and that leads to chronic inflammation. Your immune system is like the military, with two main branches: the innate immune response.
So, Vishal, can you really activate your natural healing system?
Immune response. The innate immune response includes cells that are nonspecific, meaning that although they distinguish an invader from a human cell, they don't distinguish one invader from another invader. The innate response is also feverishly fast, working within minutes to hours. Get it? Feverishly? That's because it's responsible for causing fevers. The trade-off for that speed is that there's no memory associated with innate responses. In other words, the innate response will respond to the same pathogen in the exact same way, no matter how many times it sees the pathogen. The innate immune response includes things that you might not even think of as being part of the immune system. Things like chemical barriers, like lysozymes in the tears, and a low pH in the stomach, as well as physical barriers, like the epithelium in the skin and gut, and the cilia that line the airways to keep invaders out. In contrast, the adaptive immune response is highly specific for each invader. The cells of the adaptive immune response have receptors that differentiate one pathogen from another by their unique parts, called antigens. Adaptive immunity is also diverse, meaning it can recognize almost an infinite number of specific antigens and mount a specific response against each of them. The trade-off is that the adaptive response relies on cells being primed or activated so they can fully differentiate into the right kind of fighter to kill that pathogen, and that can take a few weeks. But the great advantage of the adaptive immune response is immunologic memory. The cells that are activated in the adaptive immune response undergo clonal expansion, which means that they massively proliferate. And each time the adaptive cells see that same pathogen, they massively proliferate again, resulting in a stronger and faster response each time that pathogen comes around. Once that pathogen is destroyed, most of the clonally expanded cells die off, and that's called clonal deletion. But some of the clonally expanded cells live on as memory cells, and they're ready to expand once more if the pathogen ever resurfaces. Now it's time to meet the soldiers, which are the white blood cells or leukocytes. Hematopoiesis is the process of forming white blood cells, as well as red blood cells and platelets, and it primarily takes place in the bone marrow. Hematopoiesis starts with a multipotent hematopoietic stem cell, which can develop into various cell types. Its future is undecided. Some become myeloid progenitor cells, whereas others become lymphoid progenitor cells. The myeloid progenitor cells develop into myeloid cells, which include neutrophils, eosinophils, basophils, mast cells, dendritic cells, macrophages, and monocytes, all of which are part of the innate immune response and can be found in the blood as well as in the tissues. The neutrophils, eosinophils, and basophils are considered granulocytes because they contain granules in their cytoplasm, and neutrophils in particular are also referred to as polymorphonuclear cells, or PMNs, because their nuclei contain multiple lobes instead of being round. During an immune response, the bone marrow produces lots of cells, many of which are neutrophils. Neutrophils use a process called phagocytosis. That's where they get near a pathogen and reach around it with their cytoplasm to swallow it whole, so that it ends up in a phagosome. From there, the neutrophils can destroy the pathogen using two methods. They can use their cytoplasmic granules or oxidative burst. First, the cytoplasmic granules fuse with the phagosome to form the phagolysosome. The granules contain molecules that lower the pH of the phagolysosome, making it very acidic, and that kills about 2% of the pathogens. Now, the neutrophil doesn't stop there. It keeps swallowing up more and more pathogens until it's full of pathogens, and at that point, it unleashes the oxidative burst. During an oxidative burst, the neutrophil produces lots of highly reactive oxygen species like hydrogen peroxide. These molecules start to destroy nearby proteins and nucleic acids within the phagolysosomes, which are the components of the pathogen that has been ingested. The net result is that the pathogen is eliminated. Now, in comparison to neutrophils, eosinophils and basophils are far less common. They both contain granules that contain histamine and other pro-inflammatory molecules. Eosinophils stain pink with the dye eosin, which is where they get their name. They are phagocytic cells, even though it's not their primary mechanism of attack. They are best known for fighting large and unwieldy helminthic parasites, or worms, by releasing molecules that can poke holes in the outer layer of helminths. These cells are also involved in allergic reactions such as atopic dermatitis and allergic rhinitis, also known as hay fever. When involved in allergic reactions, eosinophils degranulate, meaning they release various enzymes and proteins within their granules an inflammatory reaction. Next, you have basophils, and they stain blue with the dye hematoxylin. And unlike neutrophils, basophils are non-phagocytic. On the flip side, they have granules that contain histamine and other pro-inflammatory molecules. Therefore, they are important in initiating allergic responses. Finally, there are the mast cells, which live in tissues, not in the blood, and are very similar to basophils. They are also non-phagocytic and are involved in allergic responses. Next up are the monocytes, macrophages, and dendritic cells, which are also phagocytic cells. They gobble up pathogens, present antigens, and release cytokines, which are tiny molecules that attract other immune cells to the area. Monocytes only circulate in the blood. Some monocytes migrate into tissues and differentiate into macrophages, which remain in tissues and aren't found in the blood. Dendritic cells are the prototypical antigen-presenting cell. Dendritic cells are usually found in sites that are in contact with most external antigens, like the skin epithelium or the gastrointestinal mucosa. When dendritic cells are young and immature, they're excellent at phagocytosis, constantly eating large amounts of protein found in the interstitial fluid. But when a dendritic cell phagocytoses a pathogen, it's a life-changing, coming-of-age moment. Mature dendritic cells will destroy the pathogen and break up its protein into short amino acid chains. Dendritic cells will then move through the lymph to the nearest lymph node, and they'll perform an antigen presentation, which is where they present those amino acid chains, which are antigens, to T cells. Antigen presentation is what connects the innate and adaptive immune systems. Antigen presentation is something that can be done by dendritic cells, macrophages, as well as monocytes, which is why all of these cells are referred to as antigen-presenting cells. Dendritic cells are the best at this process because they are the only cells that live where pathogens enter, through the epithelial like skin, gut, and airways. And they are the only cells that can traffic from these tissues to lymph nodes, where T cells circulate. Now, only T cells with a receptor that can bind to the specific shape of the antigen will be activated, and that's called priming. It's similar to how a lock will only snap open when a key with a very specific shape goes in. However, T cells can only see their antigen if it is presented to them on a silver platter. And on a molecular level, that platter is the major histocompatibility complex, or MHC for short. So the antigen-presenting cell will load the
I am
antigen
not
on an
audible.
MHC molecule and display it to T cell.
Sorry. Hello.
Ma'am, I am not audible to this video.
Oh, you can't hear the audio? Okay.
No, ma'am.
Okay.
cells. And when the right T cell comes along, it binds. The final group of they are the only cells that can traffic from these tissues to lymph nodes, where T cells circulate. Now, only T cells with
Don't overwhelm yourself, guys, that what are the information present in this video. We don't know these terminologies. So this is just an overview. You have heard these terms like antigen, macrophages, neutrophils, eosinophils in a very descriptive way, right? So this is just a very good summary of where these puzzle pieces are fitting. Okay, so just take this as a very informative video. Don't dwell too much that, oh, I don't know this, I'm not aware of that, or oh, I have not read this. Okay, all these things will be dealt in a very precise way in the coming sessions. Okay, just understand this video in a way that you are dealing with the immunology subject. So what are the things that make the immune system, where they are fitting, just like that. Okay, so just take it very lightly.
receptor that can bind to the specific shape of the antigen will be activated, and that's called priming. It's similar to how a lock will only snap open when a key with a very specific shape goes in. However, T cells can only see their antigen if it is presented to them on a silver platter. And on a molecular level, that platter is the major histocompatibility complex, or MHC for short. So the antigen-presenting cell will load the antigen on an MHC molecule and display it to T cells. And when the right T cell comes along, it binds. The final group of blood cells, the lymphocytes, includes B cells, T cells, and natural killer cells. B and T cells make up the adaptive immune response, while natural killer cells are part of the innate immune system. B cells and natural killer cells complete their development where they started, in the bone marrow, whereas some lymphoid progenitor cells migrate to the thymus where they develop into T cells. All of the lymphocytes are able to travel in and out of tissue and the bloodstream. Natural killer cells are large lymphocytes with granules, and they target cells infected with intracellular organisms, like viruses, as well as cells that pose a threat, like cancer cells. Natural killer cells kill their target cells by releasing cytotoxic granules. These granules contain molecules that punch holes in the target cell membrane by binding directly to the phospholipids and creating pores, and release some molecules that get inside the cell and cause target cells to undergo apoptosis, which is a type of programmed cell death. B cells, like T cells, also have a receptor on their surface that allows them to only bind to an antigen that has a very specific shape. The main difference is that B cells do not need antigens to be presented to them on an MHC molecule. They can simply bind to an antigen directly. When a B cell binds to a protein antigen that's on the surface of a pathogen, it's capable of internalizing that antigen, degrading it, and presenting it to T cells. So technically, they're also antigen-presenting cells as well. Like other antigen-presenting cells, the B cell loads the antigen onto an MHC molecule called MHC2 and displays it to T cells. When a T cell gets activated, it helps the B cell mature into a plasma cell, and a plasma cell can secrete lots and lots of antibodies. Typically, it takes a few weeks for antibody levels to peak. The antibodies, or immunoglobulins, have the exact same antigen specificity as the B cell they came from. Antibodies are just the B cell receptor in a secreted form. In the plasma, which is the non-cellular part of blood, attaching to pathogens and tagging them for destruction. Because antibodies aren't bound to cells and float freely in the blood, this is considered humoral immunity, a throwback to the term humors, which refers to body fluids. Now, the final type of lymphoid cell is the T cell, and it's in charge of cell-mediated immunity. T cells are antigen-specific, but they cannot secrete their antigen receptor. A naive T cell can be activated or primed to allow it to turn into a mature T cell by any of the antigen-presenting cells, but most often it's done by a dendritic cell. Now, there are two main types of T cells, CD4 T cells and CD8 T cells, where CD stands for cluster of differentiation. There are hundreds of CD markers in the immune system, and these CD markers are useful in telling different cells apart. For example, all T cells are CD3 positive because CD3 is part of the T cell antigen receptor. So CD4 positive T cells are actually CD3 positive, CD4 positive. And these cells are called helper cells because they're like generals on the battlefield. They secrete cytokines that help coordinate the efforts of macrophages and B cells. Helper T cells can only see their antigen if it's
Yes.
Ma'am, why is it saying that T cells can't secrete antigen-specific receptors?
Because the role of T cell is to kill the infection at the end of the day, right? But because the type of receptor T cells have, so for example, if you are talking about CD4 plus T cells, okay, so CD4 T cells are called helper T cells. As the name suggests, that they'll help someone else to do the job, okay. So in this case, CD4 basically helps B cells which secrete the antibody, okay. So okay, I have to go back to the presentation, just give me 1 minute. So these are the roles of where's the presentation? So this one. So if you can see it here, this. So every single cell in the immune system have their own functionality. So okay, let me think of a better example. So imagine you have a class of 20 people, right? All those 20 individuals are different in their own way, right? Not all 20 of them will be same, correct? But to organize them, you must have a class monitor, okay. So the class monitor will basically take care of whatever is going on between these 20 individuals. So if there is a conflict between them or any issue is going on between these 20 individuals, the class monitor will take care of it. But do you think that the class monitor has all the power what a class teacher have? No, right? So the class monitor has limited functionality to control these 20 individuals. Now if the class monitor is not able to control them, what will he do? He'll go to the class teacher. Now the class teacher has all the functionalities which are definitely more than the class monitor, right? So the functionality of T cells, say helper T cell, if we say, the role of helper T cell is to identify the cell which has the microbe and directly try to kill it. It won't secrete its receptor or the T cell receptors to send it to the blood, and then in the blood it will find the microbe and kill it. No, because this is not the part of humoral immunity, you remember? This is the part of cell mediated immunity. That is why the cell itself needs to take part in somehow creating or helping the cells which are killing the microbe. The only cell which secretes the antibody is the B lymphocyte, and the B lymphocytes are the part of humoral immunity. The receptors of B cells can be secreted, can be made secretory after gene like manipulation, and then they become the antibody. So the B cell, when they had it on the surface, it is called B cell receptor. Once it is in the blood, these receptors are secreted in the blood and then they are called antibody. That is the function of B lymphocyte. T helper cell will help B cells to secrete these antibodies. Cytotoxic T cell will directly kill the infected cell. Regulatory T lymphocyte, whatever has been done, so the CD8 positive cells are more, CD4 positive cells are more. Now there has to be a feedback system as well, right? Because the infection is gone now. So there has to be some other cell which will reduce the things which were secreted while there was infection. So that is the role of regulatory T lymphocyte. Similarly, this is your class monitor. He can directly go to the student who is creating nonsense in the class. He can either write its name to the board or can punish it. And even if it is not accepting what the class monitor is saying, what this helper T cell will do, it will go to the class teacher and say ki, see, this is the fellow which is creating some issue, so these cytokines are the information transferring. So the cell is basically saying ki, see, this particular individual is creating nonsense, so we need to get this individual out of the class. So it is just helping. It cannot directly restigate the student because the helper T cell doesn't have that role. The job role is not there. But imagine if helper T cell is not there, the class monitor is not there, will the class teacher will ever know who was the individual who was creating nonsense in the class? No, right? So the helper T cell is helping the B lymphocyte to help cure the infection. If helper T cell is not there, it won't be able to identify the infection and cure it. In a very layman way I am saying this. There are other things also which are into play, but if you just want to understand, this is the crux of it. Okay? And these things will become more clear in the coming classes for sure. Okay?
On an MHC2 molecule. CD8 positive T cells are CD3 positive, CD8 positive, and they're called cytotoxic T cells because they kill target cells. Really similarly to how natural killer cells do it, with one major difference. CD8 positive T cells only kill cells that present a specific antigen on an MHC1 molecule, which is structurally similar to the MHC2 molecule, whereas natural killer cells aren't nearly as specific in who they kill. So now let's go through a complete immune response with the bacterial pathogen in the lungs. To start, the bacteria will have to get breathed in, slip by your nose hairs, past the cilia in the airways, and will then have to penetrate past the epithelium layer of the lungs. Once it's in the lung tissue, the bacteria will start to divide and might encounter a resident macrophage in the lung tissue, which will ingest the bacteria and start releasing cytokines. Those cytokines start the inflammatory process by making blood vessels leaky and attracting nearby eosinophils, basophils, and mast cells, which release their own cytokines in granules, amplifying the inflammation. The blood, as well as fresh new ones from the bone marrow, dive into the tissue and join the battle. If the pathogen was a virus, natural killer cells would help destroy the infected cells at this point. This is all part of the innate immune response. Around this point in the infection, immature dendritic cells residing under the epithelium digest the pathogens and move from the lung tissue over to a nearby lymph node where they present the processed antigen on an MHC2 protein to a naive T cell. The dendritic cell, which is part of the innate immune response, bridges the innate and the adaptive immune responses when it presents the antigen to the T cell, which is part of the adaptive immune response. Sometimes, if the infection is spreading, bacteria might find its way to a lymph node without the help of a dendritic cell. In this case, B cells, part of the adaptive immune response, might directly phagocytose the bacteria and present it to a naive CD4 positive T cell. Either way, if the antigen is the right fit for the T cell will begin to differentiate and undergo clonal expansion. Differentiated CD4 positive T cells will release cytokines, that will induce B cells to differentiate into plasma cells, which secrete antibodies that will go into the lymph and then into the bloodstream. The antibodies will tag pathogens, making it easier for the phagocytes to eat them. Some cytokines will activate macrophages to kill bacteria that have been phagocytized, but cannot be killed by the macrophage alone unless it gets help from its friends, the T cells, of course. If the pathogen was a virus living and replicating in the cytoplasm of the infected cells, the CD8 positive T cells would kill any infected cells that express the viral antigen on an MHC1. Over time, as the invading pathogen dies off, most of the B and T cells die of neglect, but a few turn into memory B cells and memory T cells, which linger for years in case they're needed in the future. All right, as a quick recap, the immune system has an innate and an adaptive response. The innate immune response is immediate but nonspecific and lacks memory, whereas the adaptive immune response is highly specific and remembers everything, but it takes several days to get started and almost 2 weeks to peak.
Till here, this was the very brief overview of immune system. Okay, till here, if you have any confusion, and by confusion I don't mean that, oh, how come this is working like this or that is working like that. No, by confusion I mean what is innate immune system, what is adaptive immune system, what are the components in adaptive immune system, and what are the components in innate immune system. If you have confusion in that, please let me know. And if not, and even if you are not sure, then obviously this is a very intensive subject, so with time you will be very much okay with these terms, so don't worry too much about it. The only thing which you have to do is stay consistent with the sessions. Okay, so yep, let me know what are the things which are still little bit confusing to you, and please unmute yourself. There is little bit of disturbance from one of you guys. Yeah, anyone? I can't understand. Can you little bit be louder, please?
Again, 1 second, the question can be repeated, no?
Should I repeat the question? So the question is, if I say what is a cytotoxic T cell, are you able to connect it to the branches of immune system, whether is it innate or adaptive? If yes, you are well versed with the terminologies of the things which are introduced to you till now. Okay, which is an okay growth.
Okay, ma'am.
Okay, but if you have confusion between what is innate immune system, what is adaptive immune system, then that is a problem, and we need to spend more time in the basics, because from here onwards because the players have been introduced to you. Now we'll just understand the functionality of these players in the system. So if you guys are okay with it, we can move ahead. If not, then maybe in the next class we can spend half an hour again on the basics. Okay, but for today's class, now I'm gonna switch to questions, because we have not seen some model questions which might help you in solving your assignments. Okay, so theory will keep growing, and it will be obviously continue till the course is going. So I'm gonna proceed to the questions, and these things, don't worry, we're gonna discuss in the next class. Okay, so how I basically take the questions is, if the question is little bit simple and very like one word, something like that, then I give 1 minute to the participants to type the answer in the chat box, and then I reveal the answer. Second, if the question is little bit big or lengthy, then I give 2 minutes to the participants. Generally it was 5 minutes, but 5 minutes become too much with the restricted time we have. So 2 to 3 minutes for the longer questions, and then we discuss the answer. Okay, and I'll tell you when the time starts and when the time ends. All of you, please participate. Even if the answer is wrong, it's okay, but try to participate. And another suggestion which I give is, even if you know the right answer, you should also know why the other answers are wrong. Okay, so here is the first question. So the first question says, hematopoietic, and these questions are the ones which have already been, the topics have already been discussed in these 2 weeks. Okay, so it's not anything from outside the things which have been discussed in the sessions. So the first question says, hematopoietic stem cells originate in A. bone marrow, B. spleen, C. liver, and D. fetal tissue. Your time starts now. So it's an easy question. You guys have 1 minute to answer this, and you can type your answer in the chat box. So the hematopoietic stem cells originate in bone marrow, spleen, liver, or fetal tissue. It's a pretty simple and direct question. Try to remember what are hematopoietic stem cells and from where the stem cells are generally originated. Okay. And your 1 minute is up. Let's see how many of you have participated. Okay, 4 of you said bone marrow. Let's see what is the right answer. The right answer is fetal tissue. The bone marrow is a very obvious choice, but remember, here we are saying the hematopoietic stem cells. These cells originate from the fetal tissue, and then because they are pluripotent, right, then they go into the bone marrow and there they reside, and then they, from there they completely become like multipotent, and then they distinguish into lymphoid origin or myeloid origin. Okay, this, don't worry about the cytokines here, don't worry about it. These are just the signals which are given to the cell which is there, so that it can further differentiate into that lineage. Okay, so this is just for your reference. Don't worry about these, as I've already said, but you should remember that when hematopoietic stem cell is there, it is divided into, differentiated into common myeloid progenitor cell and then lymphoid progenitor cell. The myeloid progenitor cell further differentiates into megakaryocyte, erythrocyte, and myeloblast. The megakaryocyte, at the end of the day, they the last differentiation is thrombocytes, erythrocytes is the final differentiation, and myoblasts are further differentiated into your basic basophils, neutrophils, eosinophils, monocytes. Okay, and then the lymphoid originator, they give rise to lymphocyte cell, and the lymphocyte cell differentiates into B lymphocyte and T lymphocyte. Okay, so just remember this: hematopoietic stem cell, multipotent; myeloid progenitor cell and lymphoid progenitor cell. Myeloid origin will be erythrocyte and myeloblast, or say erythrocyte, basophil, neutrophil, eosinophil, monocytes. Lymphoid progenitor cell will be B lymphocyte and T lymphocyte. Okay, coming to the next question. Lymphoid progenitor cells do not give rise to which of the following cells? Dendritic cell, lymphocyte, granulocyte, or all of the above. We just saw this, right? But still, you guys have 1 minute. Clearly think what the question is asking and then answer. So the question says, lymphoid progenitor cells do not give rise to which of the following cells? Dendritic cells, B lymphocytes, granulocytes, or all of the above. Your time has started and you'll have 1 minute. It's a small question. Everyone, please mute yourself. Just check your mic once. Okay, so the 1 minute is over. So the right answer is what? Oh. C, and Raman said A. So most of you said granulocytes, and obviously the right answer is granulocyte because this is what we saw here, right? So lymphoid tissue, lymphoid cells, are giving rise to B cell and T lymphocytes. Okay, not the granulocytes. Granulocytes will be the granule containing cells, or the basophil, eosophil, neutrophils. They are of myeloid origin. Okay. Then the next question. Which of the following cells form the innate immune system? You will have 1 minute for this, and your options are B cells, T cells, macrophages and NK cells, both A and B, or none of the above. Okay, and your 1 minute starts now. The question is very simple. It is asking you which of the following cells are part of innate immune system. And if you know the right answer, you should know why the other options are wrong. Okay. Your time is about to be done for this particular question. It's a pretty simple question. I guess most of you might get the answer. So everyone should participate in the quizzes. Okay, no one knows that whether you are right or wrong. It will help you understand whether you got the concepts right or not. So your time is up. So the right answer is, let's see how many of you gave. Like, okay, B. Okay, I'm happy with the participation. Nice. So the right answer is obviously B. So macrophages and NK cells are the part of innate immune response. B cell and T cells are the part of adaptive immune response. Okay, great. And this is just the representation of it. So NK cells are the bridge between the innate immune system and the adaptive immune system. Okay. Coming to the fourth question, as you can see, this is a bit of a lengthy question, so I'll give you guys 3 to 5 minutes for this particular question. Okay, so don't worry, read very peacefully. So you basically have to match the type of cell to their function. So the types of cells you have is macrophages, dendritic, neutrophils, basophils, eosophils, and mast cell, and the functions are killing of antibody coated parasites, antigen presentation, phagocytosis and activation of bactericidal mechanism, antigen uptake in peripheral sites, this is basically APC, release of granules containing histamine and active agent, promotion of allergic responses, and augmentation of antiparasitic immunity. And these are your options. As you can see, there are like 2 to 3 answer per match the following. Okay, at least for a few options. So take your time. I'm giving you guys 5 minutes. Use your notebooks and tell me the right answer for this. And your 5 minutes starts now. your 3 minutes are over. I guess this is good enough time, but okay, let's take 1 more minute, and I want every one of you to at least attempt this question, because this will help you understand what are the roles and the functions of these important cells which are taking part in the immune response. I guess we can get back to a solution and an answer. Let's see how many people have tried. Okay, few of you have attempted. Good to see that. And it's a mixed response. I agree, this is a little bit of lengthy question, but it will be easy. Let's see what is the right answer first. So excuse me, the correct answer is B. So kudos to every one of you who have said B. So what B says is basically for the macrophages, what are the roles of or the function of macrophage. So firstly, the macrophage is an anti-presentation cell. We know this, okay, it's very well known that macrophages are the APCs, correct? And then they also play a role in phagocytosis and activation of bactericidal mechanisms, correct? Then dendritic cells, on the other hand, dendritic cells are also antigen presenting cells, right? Both macrophage and dendritic cells are antigen presenting cells. As well as the unique function of dendritic cell is antigen uptake in peripheral sites. Why? Because dendritic cells are the one which are connecting the innate immune system to the adaptive immune system, right? Then the neutrophils. So neutrophils are the primary function is the phagocytosis and activation of bactericidal mechanism, correct? Then for basophils, they are promotion of allergic responses and augmentation of anti-parasitic immunity. So wherever basophils are there, you should remember that there will always be an association of anti-parasitic immunity, okay? Then eosinophils, they basically killing of antibody coated parasites. So eosinophils kill the parasite once it is coated with antibody, okay? Then last but not the least, mast cell. Again, mast cells are known to contain histamine, okay? So wherever there is mast cell, you just look into the answers, and wherever histamine is there, and obviously you cannot fill it like a blind person, but most of the time the functionality of mast cell is by their ability to release histamine, okay? So this will help you understand what are the roles, and you can see the roles are also overlapped. So one cell, if it is performing phagocytosis, it is also an APC or antigen presenting cell, okay? Coming to the next question. So I'll give you guys say, okay, we'll first read the question. So what is the function of regulatory T cells? First, to kill other cells that are infected with virus or other intracellular pathogens bearing the antigen. To provide signals, often in the form of specific cytokines that activate the functions of other cells. To suppress the activity of other lymphocytes and help to limit the possible damage of immune response, or all of the above. I'll give you guys 2 minutes for this question, okay? Try to understand what this question is asking, which is pretty straightforward, and go through each and every option very thoroughly, okay? And your time starts now. Your 1 minute is over. Everyone please try attempting these questions. 1 more minute to go. Excuse me. So your 2 minutes are over. Let's see how many of you at least attempted this question. So okay, good enough. But I want every one of you to at least try attempting, okay? Even if the answer is wrong, it's okay. Anyways, we're gonna know what is the right answer, okay? So the correct answer is C. So as you remember when we were discussing about the roles of different cells, so few cells are involved in helping other cells, few cells are involved in directly killing the cells, but what type of cells are involved in regulating and making sure that the homeostasis is maintained? That is the sole responsibility of regulatory T cells, okay? So the function of regulatory T cell is to suppress the activity of other lymphocytes and help to limit the possible damage of immune responses, okay? This is the basic explanation for this. You guys, I'm gonna share these slides as well as gonna upload the live sessions. You can read it at that time as well, okay? So coming to our next question, circulating lymphocytes encounter antigens in. Now here we are going a little bit deeper into the functionality of cells, right? So first, what are circulating lymphocytes? You should know them and where they are present, and then you can very easily answer this question. So the question says, circulating lymphocytes encounter antigens in primary lymphoid organ, peripheral lymphoid organ, both A and B, or none of the above. So I'm gonna give you guys 1 minute for this question and your time starts now. Okay, your 1 minute is up. Let's see how many of you answered. At least 2 people answered. Please guys, try attempting it. Even if it's wrong, it's completely fine, right? So the right answer is B, peripheral lymphoid organs. As the name suggests, circulating lymphocytes, they'll be in the peripheral lymphoid organs, because primary lymphoid organs are associated with activating or adapting or making or maturing the lymphocytes, okay? So the next question for today is, which cells are present in the marginal zone of the spleen? It's a easy question. You will have 1 minute for this. So remember, the question is saying marginal zone of the spleen. You should know what type of organ is spleen and which zone is the marginal zone. So what cells are present there? T cells, B cells, macrophages, or all of the above. Your time starts now. I hope you guys have at least attempted. Good to see that you guys are attempting. So the right answer is all of the above, right? Because this is the proximal end of things, this one, and you will get majorly BT cells here, T cells here, and B cells here. If you're following the lecture, you know what this means, and I'm not gonna go deep into this. In the next class, we're gonna understand the secondary lymphoid organs. Okay, so with the lack of time, we still have 2 to 3 questions to go, so I'm gonna go a little bit quickly. Okay, so Peyer's patches are a part of, so you will have 30 seconds to answer this. Your time starts now. So what are Peyer's patches? You should know it is a part of first week's lecture. So is it a part of Galt, Malt, Balt, and or Nalt? Okay, try attempting it. Okay, your time is up. So the Peyer's patches are a type of gut associated lymphoid tissue. This is basically the link of a video which will tell you exactly how the Peyer's patches look like and what is their function. Okay, then it's a little bit bigger question, so we'll gonna go with our 5-minute time limit. So the question says, identify the correct sequence of events. So basically you have to summarize whole of the immune system which you just studied. Okay, go through each option very well and then try attempting it. If you can answer this, you have good enough understanding of the immune system, and we can always fill in the blanks of the things which are missing. Okay, and your 5 minutes or say 3 minutes starts now. Go through one by one option, and with your understanding try to connect whatever you have learned till now. Thirty seconds to go. Try attempting it. You will understand where are the knowledge gaps in your understanding. Okay, so anyways, we're gonna summarize things again and again, because the subject becomes very complex if you don't summarize it regularly. So I'm gonna do that, but yeah, try attempting this question. So your time is up. Let's see how many of you at least tried attempting it. Only one person, guys. Okay, so the right answer is A. So what A says, so the antigen will come and it will be presented by the antigen presenting cell. Till here, I guess you know that this is the first step. So both C and D options are wrong. Only A or B can be the right answer. Second is recognition of antigen by lymphocyte. Okay, so even here till A and B option are right. Next, differentiation into lymphoblast. Okay, or division into daughter cells with identical specificity. So if you remember that myeloid differentiation or clonal selection we spent a good enough time, so there you will have a lymphoblast which will further undergo clonal selection. Okay, I guess most of you were confused with this part itself, and the confusion came because you need to be updated with the lectures. Okay, I'll take that clonal selection topic again in the future if you guys have problem. So division into daughter cells with identical specificity, and then finally differentiation into effector cells. And even if you guys had a problem in choosing the right answer, you know the starting point is APC and the ending point is the effector cells, the cells which are actually showing the effectors. So with this, only the A option would have been the correct answer. So even if you don't know the exact answer, you can always, based upon your limited information and knowledge as well, you can choose the right answer. Okay, so for today, the last question is, B cells mature into antibody producing cells in B cell corona, marginal zone, germinal centers, or red pulp. It's a easy question. I want every one of you to answer this question. Obviously the right answer is germinal center. This is what we have seen, right? That when cells mature after they receive signal, all this is happening in the secondary lymphoid organ, and even in the secondary lymphoid organ, the germinal centers. Okay, so these are questions. So we're gonna discuss all these questions. So I have more questions to discuss, but due to time restriction, I'm gonna keep my lecture short for today. And I hope that you guys are able to understand little bit of things. It's a complex subject, I understand, but if you keep up with what I am discussing and what sessions are there and the lectures which are already uploaded, you will be able to get a basic understanding of immunology. Okay, so thank you so much for your patience, and I hope to see you all again in the next class. So thank you so much, guys. Thank you.